Hey guys, Miss Cote here, and this is the final video in the biochemistry unit. In the previous videos, you've reviewed the biological molecules, carbohydrates, lipids, proteins, and nucleic acids. And while we're learning about the biological molecules, this is the perfect time to take a closer look at a specific type of protein called enzymes. There are thousands of chemical reactions happening in our cells every day, and they all require enzymes. So in this episode, we'll review some basic knowledge about the structure of enzymes and how they work, and then look at how enzymes can be regulated. So let's get started. If left on its own, chemical reactions could still be able to take place. However, they would happen very slowly, if at all. To make things more efficient, there are agents that can speed things up and get that reaction going. The generic term for these agents are catalysts, and they can be either inorganic or organic. Enzymes are a type of organic catalyst. Remember, organic molecules contain carbon, and they fall into the protein category. For example, hydrolyzing sucrose into the two monomers of glucose and fructose is accomplished by the enzyme sucrase. I guess this is a good place to remind you guys of a couple things. One, if you didn't remember, if you see a word that ends in ASE, then it's an enzyme, but not all enzymes end in ASE. Second, enzymes are very specific for what they do. This enzyme only breaks apart sucrose into its two monomers. It cannot do the opposite reaction to make sucrose or work on any other molecule. And third, you'll come across a lot of enzymes throughout this course. Their names can get somewhat complicated, but usually their names will give you a hint as to what their function is. To get any reaction going, it requires an input of energy. We call this initial input of energy activation energy, or the free energy of activation. So to see a visual representation of this, here we have a substitution reaction, where AB and CD will be converted to AC and BD. And even though the overall reaction will give off energy, see how the products are lower than the reactants, this reaction still needs an initial input of energy to go to completion. Please note that this free energy and delta G stuff is discussed at the beginning of the chapter, which we haven't gone over yet. We'll deal with that when we get to our energy unit. I'm just trying to keep this simple and keep our main focus on enzymes. So I said before that enzymes can speed up a reaction, but how does it actually do that? They speed up reactions by lowering the amount of that initial input of energy, the activation energy, that's needed to get the reaction going. This is represented by comparing no enzyme, which is the black line, and having an enzyme, the red line. You're not changing the amount of free energy in the reactants or the products, just the speed at how it gets from point A to point B. Some more vocabulary for you guys. When we're talking about enzymes, instead of reactant, the term we use is called the substrate. That is the molecule or molecules we start with. The substrate will bind to a very specific region on the enzyme called the active site. Remember, enzymes are very specific for the molecule that they work on, so binding of the enzyme and the substrate need to fit together like a lock and key in order for it to work. Here we have an enzyme in blue, and there's an area with a specific shape where the substrate can bind to, the active site. Remember that enzymes are proteins, and proteins are made up of amino acids. The way this enzyme has folded and taken shape is due to the interactions between R groups. In the active site, there are certain functional groups from the R groups that are sticking out. They are able to bind to the substrate and position it in a specific way to make it easier to make or break the bonds and catalyze the reaction. This is what induced fit means. So the design of the active site is instrumental in lowering the activation energy. If you have multiple substrates, let's say you're creating a bigger molecule, making sure the substrates are in the correct position can help the reaction go faster. Straining substrate bonds can make it easier to break a polymer into monomers. Even making sure it's in its optimal condition, pH, temperature, salt concentration, etc., can help the reaction go faster. So let's review how an enzyme works. Here we have a substitution reaction, like the earlier model. The two substrates enter and fit into the active site like two puzzle pieces coming together to form the enzyme-substrate complex. Bonds are broken and reconnected while in the active site, and the substrate is converted into the products. When the reaction is finished, the products are released, and the enzyme is ready to catalyze another reaction. Note that the enzyme is not used up by the reaction. It can be reused over and over again. That's efficient because the cell doesn't need to make a lot of the enzyme, just enough to get the job done in a reasonable amount of time. Enzymes are pretty particular about the type of environment they like to work in. If the conditions are not to their liking, this can affect the rate of the reaction. Two of the biggest examples that you may remember in your honors biology class would be a change in temperature or a change in pH. Chemicals that specifically influence the enzyme will be discussed a little later in the video. Let's look at temperature first. Each enzyme has an optimal temperature where it will function best. At the optimal temperature, the protein will be in its most ideal shape and will therefore work the best. Here we have two examples: an enzyme that works in our bodies where the temperature is 37 degrees Celsius or 98.6 degrees Fahrenheit, and an enzyme that's found in a bacteria that lives in a hot spring. Notice that whatever the optimal temperature, the graphs take the same shape. Below the optimal temperature, the left side of the curve, we see a slower rate of reaction. The reaction is still going, but not as fast. Remember that temperature is the average measure of kinetic energy. The molecules are randomly moving around more slowly at lower temperatures, and it takes longer for the substrates and enzymes to come together for a reaction. Think about how sluggish you are during the wintertime. Above the optimal temperature, the right side of the curve, the line drops off very rapidly. This represents that high temperatures will begin to change the shape of the protein, eventually causing it to denature. You're losing the quaternary, tertiary, and secondary structures. And when the protein loses its shape, no reaction is occurring. Each enzyme also has an optimal pH that it likes to work at. Here we have two enzymes in our digestive system that prefer to work best in two different pHs. And again, no matter what the pH, the shape of the graph is the same. When you move away from the optimal pH, you begin to change the shape of the protein and therefore the active site, which causes it to work less effectively. Eventually, the shape changes to such an extent that it stops working. Enzymes can also have helpers or assistants. These are called cofactors. Their job is most likely to aid in the substrate attaching correctly to the active site. If the cofactor is an inorganic substance, then we just call it a cofactor. However, if the cofactor is an organic molecule, such as a vitamin, then we call it a coenzyme. Just be aware of the terminology. And with all of the chemical substances around in the cell, some of these can actually prevent the proper working of the enzyme. These substances are referred to as inhibitors. There are two types of inhibitors that you need to know about: competitive and non-competitive. And I want to explain the differences between the two by using the figures on the next slide. Over on the left in figure A, we see what normally happens with the interaction between the enzyme and substrate. Figure B depicts competitive inhibition. When there is a competition between people or animals, they are actively competing against each other for something, whether it's food, water, or even a soccer game. Here we have a chemical that is competing with the substrate for the active site. If the chemical gets there first, then it prevents the substrate from binding, and the reaction cannot be catalyzed by the enzyme. Figure C represents non-competitive inhibition. This is more of a passive-aggressive type of competition, where the chemical is not actively competing for the active site, but is sneakily binding to a site somewhere else on the enzyme. And as you can see, binding at this other site can change the shape of the active site, making it less effective. It's possible that the substrate could still bind to the active site, but the reaction may not go to completion. So while the end result is the same, the enzyme can't catalyze the reaction, the way the chemicals go about it are different. One is direct, while the other is indirect. This last section discusses how these enzymes are controlled or regulated. Regulation is a big part of this course, and we'll see examples of regulation at many different levels of biology. Our cells are pretty amazing because they are so efficient, and that efficiency is due to the control that the cell has over certain processes. The cell doesn't like to waste its energy or its resources. Later on in the course, we'll look at regulation at the gene level, but right now let's look at how the actual protein can be regulated by tweaking or modifying it. One way the cell can control a pathway is through allosteric regulation. The word allosteric means other object. Here, an enzyme is being regulated through another chemical binding to a site other than the active site. You may be thinking, this sounds like non-competitive inhibition. And yes, non-competitive inhibition is a type of allosteric regulation, but there's more to it than just that. This other object can be an inhibitor, which will prevent the reaction, or it can be an activator, which will stimulate the reaction. So let's take a look at how this can happen. Most of these allosterically regulated enzymes are made up of multiple polypeptides, so we have quaternary structure here. And these enzymes can oscillate or go back and forth between active and inactive forms. We can use the allosteric regulators to stabilize it in either the active form using an activator or the inactive form using an inhibitor. But in both, the regulator is binding to a site that is not the active site, but is changing the shape of the active site for the substrate to bind or not bind. Another type of regulation is called cooperativity. Here the book refers to it as allosteric regulation, but it doesn't quite fit the definition. Here we have a multi-subunit enzyme with 4 active sites. First, there's no other molecule. The regulator molecule is the actual substrate. And second, it binds directly to the active site, not another site somewhere else on the enzyme. Anyway, how this works is that when one substrate manages to bind to the active site, it changes the other active sites so it's easier for the additional substrate molecules to bind. I guess how they justify it as being allosteric is that the first substrate acts as the other molecule that binds at another site, making it easier for the open active sites to bind their substrates. Biology people are constantly arguing over this, so I guess I'll just wait and see what happens. Regulation brings us to a concept called feedback inhibition, which is how the cell can be as efficient as it is. In feedback inhibition, the end product of a pathway, especially when the cell makes enough of the product, can actually shut the pathway down at some point. Let's look at how this works. Here we have a multi-step pathway that converts threonine to isoleucine. These are 2 amino acids, by the way. When enough isoleucine is made, the cell doesn't want to keep making more isoleucine. That would be a waste of resources and energy. So in this case, the excess product can actually act as an allosteric inhibitor and bind to another site on the enzyme. This changes the shape of the active site and prevents the enzyme from doing its job, thereby shutting off the pathway. Note that in this example, we're shutting off enzyme 1, but this could happen at any point in the pathway. For example, we could shut off enzyme 3. When levels of isoleucine are getting low, the molecule acting as the inhibitor can leave the allosteric site, which turns on the enzyme, allowing it to restart the pathway again. I know we haven't gotten to the cell chapter yet, but hopefully you remember that a eukaryotic cell is not just one big bag of enzymes. They have organelles that create different compartments in the cell, so enzymes can do their jobs in specific places and not interfere with other enzymes. For example, most enzymes involved in cellular respiration are located in the mitochondria, or enzymes for photosynthesis are located in the chloroplast. As we learn about different processes important to life, we'll look deeper into the different organelles and where these enzymes actually work. So that wraps up what you need to know about enzymes and our unit on biochemistry. If you have any questions about this episode, make sure you remember to ask questions at the beginning of class. Otherwise, go out and be a good human.